Good morning, everybody. Today I want to be talking to you about the application of DNA-encoded library on RNA targets. I don't want to waste a lot of time convincing you how important it is to target RNAs. I will simply acknowledge that in the recent years there have been elevated interest in different modalities in targeting RNA, and the small molecule liganing has become a very hot topic. And in the past decade, we have seen a slowly but steady accumulation of successes in identifying small molecule ligand to RNAs. And we also started, just like we did in the protein world, we started to, try to understand the pocket that RNA can provide and how easy or how hard it is to identify small molecule ligand. And depending on who you ask, some might say that so far there are many successes or anecdotal successes. And it is obvious to us that there hasn't been a systematic way of identifying or comparing small molecules that appear to be RNA ligand. But if we take a look at what has been quite mature in protein ligand discovery, and we have a pretty large toolbox. Range from high throughput screening, fragment-based, virtual screening, structure-based, and all these tools are contributing to the small molecule identification, as well as the DNA-encoded library. And we seem to convince, or at least have to believe, that the identification of a small molecule against the RNA target is somewhat similar to proteins. So there are a lot of thoughts how to apply these technologies in identifying those ligands for RNA. So obviously, the DNA-encoded library as an affinity-based screening technology, we often get asked that, can you apply DEL to RNA targets? So if you look at this problem at the face value, you will be concerned, obviously, because the DNA-encoded library, it is the barcode is a DNA that poses some challenges against RNA targets in at least two folds. First, the barcode itself is negatively charged, and the RNA is also negatively charged. So there might be a electrostatic repulsion between the two molecules. And the other concern is there might be RNA-DNA interaction that may contaminate your screening. To give you a little bit of more detail on this, and the mainstream DNA-encoded library deploys a double-strand DNA, but there is a single-strand tail. As shown here on this graph, the purple region is a single-stranded DNA that contains a unique molecule identifier, which is an N12 randomized sequence. The reason that we have that is that it allows a better amplification efficiency for the DEL molecules, and it is a pretty common practice. So if you think about it, you have a single-strand DNA in there that must be a high risk that it might hybridize with your RNA targets. So if you can ask any molecular biologist, they will say, well, this is very simple. You just do a primary extension to cover up this region, and now it is a double strand. That you might mitigate this problem. But is that the case, right? We don't know. So we decided to design an experiment to answer these two questions. We picked an RNA target, in this case, THF riboswitch. And the reason we picked that is it has a well-known ligand. The ligand is chemically accessible to add a DNA tail. Okay, so we made an on-DNA version of the THF, and we ligate it up such that it looks like a real DEL molecule that has also the randomized single-strand sequence. And then we asked if this tool compound will still bind to the riboswitch that is presented on the solid surface. The answer is clearly yes. The on DNA compound does bind to the rebel switch. Okay, that basically answers the first question, that the electrostatic repulsion is not too much of a concern. At least it serves as a selection stringency control, right? Any strong binder that can fight against this repulsion force will be the ones that we're looking for. And so we also compared the blocked version and unblocked version. The blocked version meaning the single strand region of the del molecule is masked by another oligo. So as you can see clearly here, without blocking, the negative control and the tool compound appear to be the same. They all bind to the rebel switch to some extent. But if you block them, the signal difference is quite big, and then that is very comfortable for del selection. And we also looked into the we wanted to be sure that this is really due to the single-stranded region. So we developed our own motif analysis that we looked at all the sequences that we enriched from this experiment. As you can see, the top enriched sequences, they actually is exact complementary to the A region shown here, the loop region. So that confirms our concern that yes, the single strand region is responsible for this background binding. And if we look closer to look at all the motifs that we discovered from this approach, we found that they can actually map to several regions. And if you look at the structure of the rebel switch itself, and actually they concentrate on three regions. One is the red region I already talked about, and then the other is the gray and the blue. So it is very clear to us that the single strand region of the del molecule actually will find the loop region of a rebel switch. Okay, but the good news is if you block it, you can pretty much eliminate all these background binding. So with these two experiments, we are convinced that yes, there are challenges in applying del technology to RNA targets. However, these challenges are manageable, right, via molecular biology and a little bit of informatics. So next, I wanted to give you a real case. We wanted to give us a challenge to find a real RNA target, and we set our eyes on RNA G-Quadruplex. This is a simple structure, but have a very high occurrence in a genome. Although the function of it is still debatable, there are many proposals. But for us, as a platform to generate the toolbox for a target class, I think this is great. Right? So we wanted to focus on this region, this G-Quadruplex. Actually, we set up a little bit of challenge that we want to just screen against this region. It is very small. It possesses very simple pocket. And we'll see if it is too simple for us to find any hit. So we picked out a sequence from the literature. This is, you can see it's the bare minimum of the full stretch of G's. It forms a cage. And we added 4 U's of the 5' end, and we added a biotin. That gives us a very, very basic system that we can immobilize it. But to make sure that this sequence can actually form the G-Quadruplex, we give it a little bit of test. And we know there's a ligand that binds to G-Quadruplex, and then we measure the affinity of this ligand to this sequence, and it appears to have 100 nanomolar binding affinity. So that convinced us that yes, although the sequence is very short, we do have the structure present. So in the DEL selection, there are many sets of data to review, but here I will focus on two libraries that produced interesting hits. One is a 3-cycle library. The 3-cycle library means there are 3 building blocks. And as you can see here, a lot of hits, they show up in a pattern that 2 building blocks are required, and then the 3rd building block can take some variables. Another library here, the library 114, is a 2-cycle library. It consists of 2 building blocks, right? So if we make the compound proposed from the selection, we actually get decent binding to the G-Quadruplex as a target. So the first 2 actually we had trouble determining what is exact KD by SPR, so we had to call in another technology, MST, to measure its affinity. The last compound shown here is 1.7 micromolar affinity confirmed by SPR, and on MST it reports a 20 micromolar binding affinity. All right. So, because it's a short talk, but I hope I have convinced you that there are some manageable challenges in applying DEL to RNA targets. However, you can comfortably discover ligand against RNA with this technology. And for us, like I said, it's just the beginning, and we wanted to, obviously right now we only have, a couple of cases. We hope that by screening against more and more RNA targets, we'll come to understand, the pocketome of in this world. So please, if you have any RNA targets in mind, we are here to work with you. Thank you.
Any questions for our speaker?
Yeah.
Great talk.
Thanks.
Thank you. Two questions. How big was the RNA target, and with how many copies was each library member represented?
How big is the RNA target?
Yes.
The RNA target is about 20 nucleotide.
Okay. And approximately how many copies each library member was represented during that selection experiment?
So we have on average 10,000 to 100,000 copies per library member, and we typically run 2 rounds of selection. Sometimes 3 rounds. In this case, I believe it's 2 rounds of selection.
Okay, great. Any other questions? Great. Let's thank our speaker again.
Thank you.